So you found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there YouTube, the Dapper Dinosaur here. Today we're taking our final look at a talk Georgia Purdom gave sometime around the end of 2009 or beginning of 2010. This talk is about genetics, as you probably gathered from the title, and while I'd appreciate it if you watched the previous videos, you don't really need to, since she's starting a new topic right as we get into this. Additionally, because this video was uploaded in potato quality, we're watching it on my very, very real, very low resolution CRT TV. Okay, well, here's Georgia.
Does gene duplication add new genes with new functions to the genome? Again, this is one we're seeing a lot of in increasing amount.
Well, it's a weird question. It's a bit like asking if eggs can make cake batter. The answer is no, but that doesn't mean eggs aren't an important aspect of cake batter. And that people who deny the existence of eggs as a reason to say cakes are impossible aren't wrong. Gene duplication means that there are now extra copies of a gene, and so if one of them mutates such that it produces a different protein, a copy with the original sequence still exists, so the original protein is not lost. This way, the number of genes can increase without requiring previous functional proteins to be lost. It's not the only way that this can happen, but it's a very important way it can and demonstrably does happen.
Okay, the facts. Gene duplication is a duplication of a region of DNA that contains a gene. I read that definition later. I thought, well, yeah, duh. That kind of makes sense.
Yeah, of course it does. Does Georgia think that geneticists are out here trying to trick everyone? Oh, right. She actually does think that. Since I'm bad at segues...
Hey!
Something was brought to my attention by my patrons and members, and it's that Georgia has been affected by degenerative hearing loss since high school. This may explain some of her mispronunciations, although given that N is
alveolar
and M is bilabial, and she claims to be able to read lips, I'm skeptical. Probably doesn't explain conundrum though. Well, I don't give a single about Georgia's feelings, since she's a propaganda minister for a Christo-fascist and works for an organization that kills people, albeit indirectly. I do care about my audiences, so let me apologize.
What you're doing is just taking a chunk of the DNA that has a gene and copying it, so now you have two copies instead of one copy, which is what you see in the picture here. You have two blue regions instead of one.
Yep, we all get it.
Now, that's very harmful in animals and humans. In fact, it's been linked to several neurological disorders in humans, including Alzheimer's disease.
Saying it's associated with Alzheimer's disease is, well, it's not
untrue.
But saying it that way is misleading as hell. One rare form of Alzheimer's disease is associated with the duplication of the APP gene, a gene that codes for amyloid beta precursor protein. Virtually no one with Alzheimer's disease has this duplication, as it's quite rare. It's kind of like saying that cats are associated with heart attacks because some people have been found to have had heart attacks after being startled by cats. Sure, I guess that would be an association, but if I use that as some attempt to say that cats should be abolished or that they're dangerous monsters, you'd be right to ridicule me out of the room. But further, it's actually pretty easy to find instances of gene duplications that are not harmful or even beneficial. For example, dogs are a subspecies of gray wolf, and they have far more amylase genes than any other group of wolves, which helps them with their starch-heavy human-like diets. In fact, they even express amylase in their
salivary
glands. Like humans do, but unlike other canids, these are a result of a multi-gene duplication. In fact, one of the biggest genetic differences that affect the brains and cognition of humans, which has allowed them to be by far the most cognitively complex of the apes, is due to multiple duplication events involving both partial and whole gene duplication of the SRGAP2 gene. Something unheard of in other lineages of mammals. Every kind of mutation is associated with harmful effects, but that's like saying that organic molecules aren't toxic at relatively small doses. I mean, a lot of them are, but a lot of them are not. Sure, I don't want to drink a glass of benzene or eat a tablespoon of potassium cyanide, but on the other hand, it's going to be hard to poison myself with sugar.
Paget's disease of the bone, and an immunodeficiency disorder.
And probably more. Gene duplication at a basic level tends to result in more production of the gene product, and sometimes in cells that don't normally produce it. So yeah, if more of a protein is bad, then it'll be bad. But if more of it isn't bad, then it won't be. It's not really all that complex to understand. Suppose the
lactase
gene is duplicated in some group of humans who currently do not have
lactase
persistent, so they're all lactose intolerant, the ancestral condition of humans. This would mean that they produce more
lactase
and so can digest lactose later in life. This gives them a new food source, at the relatively minor cost of making it slightly harder to wean children. Or, as we already said, suppose a wolf living in association with humans had a duplication of its amylase gene, allowing it to get more calories from food scraps that humans leave behind for it. It's not hard to see that for some proteins in some circumstances more would be good, and in others it would be bad. Going through a list of instances where it's bad does not negate the times it is good. This is like going through a list of diseases caused by bacteria to show that bacteria are bad without stopping to notice that without bacteria, the earth would be uninhabitable in short order as all plants die and animals fail to be able to digest anything but the most simple foods. Like everything in biology, whether bacteria
or
gene duplication are good or bad is context dependent.
So it's been linked to lots of problems, at least in animals and in humans.
So has heat. I wonder if Georgia will stop cooking her food.
And specifically, I talked about humans there. But it's very common in plants. They have a condition called polyploidy, where they actually duplicate entire sets of chromosomes, not just genes, but whole chromosomes.
Polyploidy isn't just the duplication of chromosomes. It's specifically the duplication of the whole genome, going from diploid to triploid or higher. It also happens in animals from time to time, although it's rarer. For example, the marbled crayfish is an asexual triploid crayfish which evolved in the aquarium trade from the wild crayfish Procambarus phallax, which is native to the states of Georgia and Florida, and that's the North America Georgia, not the one in Europe.
And it's very common, but it can be very beneficial in plants in specific environments. It may even result in speciation. It may help them adapt to the environment that they're in.
The same can be said for animals. Many animal lineages show signs of whole genome duplication in the past, and the fact that the marbled crayfish is dominating European waterways means that
polyploidy
seem to be helping them quite a bit, even if it's a bit of a disaster for the other species of the rivers of Europe.
So it's not to say that gene duplication or chromosome duplication doesn't occur. It does, but again, it's only really helpful in certain kinds of organisms.
Like humans, crayfish, wolves, icefish, many plants, bacteria, etc., etc., etc. In other words, it's helpful in basically all organisms sometimes, and harmful in all of them other times, like pretty much any category of mutation. By the way, if you enjoy this video, please share it. I want as many people as possible to see just how much creationists get science wrong.
So how does this relate to evolution?
Well, gene duplication allows for subsequent mutations to occur to a gene, while leaving other copies with the original sequence intact, meaning that it is a source of new genes. And of course, just the duplication on its own can be beneficial and spread through a population, like it did in the human and dog lineages.
Well, gene duplication is proposed as a major factor in generating new genes. This is how you get new stuff.
It's one of the ways to get new stuff, and we can actually see that in the nightshade families of plants, which includes the tomato and the potato. That lineage had a whole genome duplication, resulting in having 4 copies of each chromosome, and for the most part, even today, the duplicated copies are mostly that, but in some cases,
the homologous gene in one copy actually produces something different.
This is those organisms using duplicated genes as the basis for new genes. It observably happens. Another example occurred in the Nototheniode ice fish, which had duplication events to their
trypsinogen
gene that resulted in a new antifreeze protein that allows them to inhabit the far southern waters around Antarctica.
Right? You got to get new stuff. If you're going to go from a bacteria to human, like I say, you can't just tweak what's there. You got to add stuff. You got to add information.
There's that undefined word again.
To get from one kind to another. And gene duplication provides the raw material for evolution.
I mean, that's part of what it does, but like we saw in the previous entries in the series, sometimes junk DNA can gain function too. Junk DNA that was never part of a gene. And of course, there's no such thing as a kind. Or if there is, then there exists only one of them. Kind is just a weasel word that creationists use, that no matter how much evolution they're forced to accept, they can pretend that it doesn't count, because there was no change in kind. If creationists actually had any intent of doing real science, they'd come up with a rigorous way to tell that two organisms are not in the same kind, which is to say they couldn't share a common ancestor. So far, no luck. And really, we don't even have much of a definition that we could test, meaning that since the idea of kinds is non-falsifiable, it doesn't belong anywhere near science.
That's what mutation and natural selection will act on. They will act on that duplicated material.
Well, they'll act on any part of the genome, including those parts. It's not like mutations know that some parts of the genome has been duplicated.
And just to give you, again, some quotes on this, gene duplication is the primary source of new genes.
I haven't run the numbers on that, but it doesn't seem absurd on the face of it. Also, I don't really care at this time to try to figure out if most new genes come about after gene duplication. I'm not sure why the prevalence of that source of new genes versus some other source is important to the question of common ancestry.
This is how we get new stuff. This is how evolution is possible because of gene duplication. Evolutionary biologists agree that gene duplication has played an important role in the history of life on Earth, providing a supply of novel genes that make it possible for organisms to adapt to new environments. This is how it works. The existence of diverse multi-gene families, particularly in eukaryotes, provides evidence that numerous events of gene duplication followed by functional diversification, we'll talk about that, have shaped genomes as we know them. So he says what happens is you get a duplication. You got one gene, now you get two genes. Now, mutation and natural selection act on this one, you get a brand new gene that does something new, but you still have the old one. Yay, okay? Now we can go from, bacteria to humans. That's at least the idea there.
No, because bacteria and humans share a common ancestor. Humans didn't evolve from any extant organism, not from chimps and not from bacteria, but they did evolve from a single non-eukaryotic cell. So if that's what she's trying to say by inaccurately saying bacteria, then sure, that's a big part of the process.
Over time, you're going to go from really small, simple genomes to really complex big ones like we have in human beings.
Selection can drive complexity, but really it's neutral evolution that does that for the most part. And it even acts on gene duplication. Genes are most likely to lose some function after a duplication than gain it. But if you have two copies of a gene and one loses function A, but the other loses function B, between the two of them you still have function A and B. And now you have a more complex chemical pathway to get whatever the full functionality originally was. Then later, creationists will come along and declare that's so complex and that couldn't have evolved, and say it's proof of supernatural, intelligent magic design. And take that, atheists, and so on and so on.
And some evolutionists have even suggested that whole genome duplication, it's not even portions of the genome, but you duplicated the whole thing, and that's how you get these big jumps. Because, how do you get to an invertebrate to a vertebrate? Okay, that's a big jump.
Is it? Because I'm not sure what the big jump is with adding some extra cartilage around the
notochord,
which is all the transition from non-vertebrate to vertebrate required. I mean, look at a sea lancelet.
the closest invertebrate to being a vertebrate.
I don't think that needs a whole genome duplication to get a spine. It already has most of what's in a spine anyway. And if you have all of a spine, you have the courage to hit like on this video. It will tell YouTube to not only show you more videos of mine, but also show this video to more people. It often seems like YouTube does all it can to keep channels like this from growing, so why not help me counter that?
And maybe that's what caused it.
No, gene duplications don't cause large-scale evolutionary change. They allow for it.
So how are they detecting these gene duplications? And how do they know this, okay? Because obviously this is something that happened in the past, right? They can't go back and look at it. I can't go back and look at it. Nobody can.
Actually, we can look at it as it happens in real time. That's the thing that happened with Lenski long-term evolution experiment with E. coli. But also, we can do things like look at dogs. Dogs have many more copies of the amylase gene. Like I've mentioned, since no other canines have these, and the other members mostly have the same amylase gene in the same spots in their genome, the only realistic possibility is that these are the result of gene duplication. In the case of things like tomatoes, it's pretty easy to come up with how many chromosomes of each an organism has, and you realize you get 4, not 2. In the case of the marbled crayfish, on a gene-by-gene level, it's almost identical
To Procambarus phallax.
but has 3 chromosomes on each type, so there aren't a whole lot of options left besides duplication.
Right, this is something in the past, so their presuppositions are going to play a big role in that.
Other than the presupposition that science works, not really. What will play a big role is the conclusions of previous scientific experiments and observations that will not be litigated in every subsequent test. Georgia doesn't like those conclusions, so she pretends that they're just assumed for the sake of convenience, despite not being well evidenced. It's quite the opposite. It's like when an astronomer tries to work out the orbital characteristics of a newly discovered exoplanet. They assume the value of the gravitational constant rather than measuring it, but they do that because the value has been determined with a high accuracy many times, and it doesn't need to be done again just to figure out the orbital mechanics of this new body. So while Georgia has decided to reject all of genetics, here in the real world we have experiments that show how to recognize things like common ancestry, point mutations, molecular homology, genetic variability, and both gene and genome duplication.
They mainly focus on gene families, as talked about here in the quote. So what is a gene family?
It is a group of genes that share a common ancestor gene. It's much like a human family. A human family, at least in terms of blood family, are a group of humans linked together by one or more common ancestors. They are recognized by either having a record of genealogy or by genetic testing in which a nested hierarchy of changes that occurred at different times can be constructed, which allow the relationship to be determined. This also occurs with language families. That's how we know that English and Hindi share a common ancestor, but that English shares a more recent common ancestor with Latin, and that even closer common ancestor with Norwegian, and yet a closer common ancestor with Dutch. This also happens with manuscripts, and is why we have biblical manuscript families like Byzantine and Alexandrian, and even subfamilies within them. Georgia can try to deny this, but this is the same kind of evidence we use to figure out what's even in the Bible that she reads and bases so many of her beliefs on.
Well, just like in a biological family, my sister and I look a lot alike, and we look a lot like our mom. So we can tell that we're a biological family related to one another.
I don't know, I wasn't there. So by AIG logic, I guess Georgia and her sister could have just been specifically created from mud and just happened to be similar because God felt like it. And don't give me this stuff about records of birth and their mom's memory. That's just part of the appearance of age that God gave to them when he created them.
Well, the same is true for genes. They have families too. They have similar genes. Similar genes have similar sequences and similar functions. Okay, so they're called a gene family because they all do something very similar. Not exactly the same, but similar.
Wrong.
Similar function is neither required for identification of related genes, nor is it enough to demonstrate where it exists. Now the members of a gene family often do have similar function, it's just they don't need to. Recall back to what we know of the ice fish who evolved an antifreeze protein from a gene that codes for the precursor to a digestive enzyme. What matters is the sequence of the DNA. Imagine a chain letter. This chain letter contains only instructions on how to pass it on, and a series of letters A, B, C, and D. We'll say that it's 150,000 letters long in that letter selection. The instructions are to use random.org to select a random letter, then use the same website to assign it a new value, A, B, C, or D. First, you set the generator to a random value of 1 to 150,000 to select the letter to be changed. Then you set it to 1 to 4 to figure out what the new value should be. And if the value doesn't change, that's fine. The instructions say to then pass that letter on to 2 people. Let's suppose that just for the sake of this hypothetical, everyone who does this follows the instructions. Let's further say that the letter starts with just 1 copy and goes through 12 generations after that, meaning that we have a bit over 4,000 copies of the letter when all is said and done. Remember, each time the letter is passed, there is a 75% chance of 1 letter being mutated. Similarly, the chance of the same mutation occurring in any other letter is 1 in 600,000. So we can be very confident that in 12 generations, it has not happened even once. Although that's not strictly impossible. The first thing you'll notice is that fully half of the letters will have one change that they all share, and that is not shared by the other half, which will have another change that they all share.
And in each half, half again.
So a quarter of the total will have another change that the other 3/4 do not, and so on, with only the 25% chance of no mutation confounding this. As a result, you will be able to build with high confidence a tree that shows the
descent
of these letters. The basic idea of
inherited
mutation is how we build the tree of life, the family tree of languages, the family tree of manuscripts, and the family trees within a single species when genealogical data isn't available. If you were
presented
with these 4,096 letters from the 12th generation and you did a statistical analysis of them, it would be so obvious that they descended from ancestral versions and all share the same common ancestor that to deny it would require you to simply lie or make up some scenario that is so fantastically unlikely
as
to be incredible to anyone who isn't either very stupid or very insane, or I suppose just very motivated to believe you.
And they claim that this is a result of an ancient gene duplication.
Usually that's true, but if 2 populations could also have a single gene diverge in function without duplication, it just means that that gene will no longer produce a protein with the original function. That might be fine if the function is no longer important, or it might be that some other gene will fill that need, which itself could be the thing that left that gene free to change in those lineages.
So probably the most used example of this is the human beta-globin gene cluster. And you may have heard this before. This is a pretty common argument, again, used for this. And beta-globin is combined with alpha-globin to make hemoglobin. And hemoglobin is what carries oxygen in our blood, in our red blood cells.
That's true.
So if you look at the beta-globin gene cluster, there's actually 5 genes there, at least that we're going to talk about, that have a role in basically the development of an organism in developing their hemoglobin. So the gene epsilon, the epsilon globin, is formed when an individual is an embryo, okay, during the about 6 to 8 weeks. So then after that 8 weeks is over with, basically, then the human will switch to, the baby will switch to forming gamma globin, okay? And that happens for the remainder of the pregnancy, so as they're a fetus. And then that gene gets turned off, and then delta and beta get turned on in the adult. Right after birth, those genes get turned on. So they all are combining with alpha globin, okay, to make hemoglobin, but there's different ones depending on the stage that the individual is at in life.
Sure does look like a gene family to me, but let's see how Georgia nuh-uhs her way out of that.
And as the next gene gets turned on, the previous gene gets turned off, okay? So it's sequential, it happens spatially and temporally.
Seems like it's just temporally and not spatially.
Georgia knows what those words mean, right?
And in fact, they're all on the same chromosome, and the order that they are on the chromosome is the order that they turn on in the life of an individual, which is pretty amazing.
Is it? I mean, the order being sequential is pretty neat, but I don't see why them being on the same chromosome is amazing. That is more likely if they form a protein family or a gene family than if they don't. In fact, it seems like if it were designed, it was designed in such a manner as to make it look, for no particular reason I can discern, as if it in fact evolved. If they were scattered all over the genome, that would be harder to explain in evolutionary terms.
Now, what they think happened about 200 million years ago, okay, even though no one was there to observe that.
And once again, it doesn't matter. If we're going to accept science at all, then we have to accept empiricism. And one of the underlying axioms of empiricism is that nature does not fundamentally change from day to day or place to place, which means we can gather data about the past in the present, because of things like laws of nature and cause and effect. The only way to say we can't is to say that science actually can't tell us anything at all about the present or the future either. Empiricism is either okay or it's not. If it's okay for the past too. If it's not, then science isn't okay at all.
About 200 million years ago, you had one beta globin gene, okay?
Good catch there. It certainly would not have been human. I also want to point out that this is the Triassic, so there are no true mammals, as they would not evolve until the Jurassic. Further, the groups leading to mammals like mammaliforms were all egg-laying, much like lizards today. They would have had soft-shelled eggs with pores to allow oxygen to diffuse into the egg, and that is where the embryo would get its oxygen, which is not too dissimilar to how it would do so in life. But the beta globin gene cluster isn't the only remnant of this basal reproductive method. Mammals also retain multiple genes that code for yolk proteins that are broken in mammals but perfectly functional in reptiles. And interestingly, the only remaining egg-laying mammals, the monotremes, have more functional yolk proteins than other mammals, but more broken ones than reptiles. You know, the kind of thing that we might expect from mammals who were evolving to have shorter times in the egg as a result of milk production long before live births. Indeed, monotreme eggs hatch only days after being laid, and so the young are dependent on the yolk for a very short time. After that, they drink milk, although interestingly, monotremes do not have nipples. Instead, milk is excreted directly from the skin along milk lines that fall in the same place as nipple lines in other mammals. It's almost like the entire evolution of reproductive strategy of humans is on display in the animal kingdom, you know, because it kind of is.
So it duplicated and you got two. And this one duplicated and you got epsilon and gamma. And this one duplicated and you got delta and beta. So lo and behold, you got the genes that you have today in the beta globin gene cluster. What's the problem with that? It's a logical fallacy, okay? Of affirming the consequent, okay? Once again, here we go. If gene families exist that share similar sequence and function, then gene duplication of a common ancestral gene must have occurred.
I bet Georgia is going to counter with untestable magic as an option.
Gene families exist that share similar sequence and function, therefore gene duplication of a common ancestral gene must have occurred.
Yawn, we're skipping Georgia's rejection of science under the guise of non-existent affirming the consequence fallacy. I've already covered it. Go back and watch a previous part for that discussion.
Something that's interesting about these gene families is, especially beta globin gene, they all have slightly different functions. They don't have the same function.
Yeah, that was part of the initial definition she gave. Is she about to say that maybe they're not a gene family because they meet her own definition too closely?
Slightly different function, but they're all doing basically the same thing, though, right? So again, it's very important to remember that, and we're going to talk about that a little bit more in a minute.
Or she could just actually finish the thought. If you ever find yourself saying,
Wheel.
Get to that in a minute, consider whether you should have just mentioned that thing you were saying about in a minute rather than just right now and cock-teasing your whole audience for no reason.
And also, here's what gets me about this that I don't understand. How do similar genes with similar functions progress you from going from molecules to man? Don't you need different genes with different functions?
Yeah, we have that too. I already gave the example of the gene for the precursor to a digestive enzyme that later became an antifreeze protein gene. The fact that Georgia is cherry-picking the gene family with extremely similar functions doesn't mean that we don't have gene families with lots of different functions.
Another example
Is the gene family that includes several kinds of bacterial
flagella.
as well as excretory systems, which are very different functions. One group of people who would never cherry-pick gene families are my
Patrons on?
Patreon. YouTube is a very fickle platform, and 1 month I might get thousands of views, more than normal, and in the next 1,000 fewer. Patreon helps me keep doing what I love, teaching science and debunking nonsense. This channel only exists because of the support of you, my viewers. The best way to support me in the long term is Patreon. That's where I get to keep the largest portion of what is sent to me. And also, it does not depend on the whims of YouTube. The Patreon starts at just $1 a month, and you can get 15% discount for pledging annually. Any pledge will get you access to my Discord servers for supporters, early access to videos like this, and there are more perks at higher tiers.
I mean, I thought that was the whole point, right?
No, there is no point to evolution any more than there is any point to electromagnetism or gravity or quantum chromodynamics. Science isn't in the business of giving us the point of things. That's for philosophers, politicians, and everyday people. Science describes what is, not what ought to be.
So how does having something similar get you to become a human being? I mean, that's very, right off the top of my head, that one really, I don't understand.
No, she understands. I have no doubt she knows she's cherry-picking, and that there are gene families that do not, in fact, all have similar functions. Also, it is the case that gene families are not the only way new genes come about. Sometimes some bit of random junk is neo-functionalized and starts a whole new gene family. This even happens at the species level in extant organisms. So it's
A post-flood thing.
according to creationist thinking. That means that if Georgia were honest with the evidence, she would have to admit that new genes can come into existence from non-gene ancestral parts of the genome. Of course, she's anything but honest with the data.
How gene families are really good proof of this, or really good evidence for this. And there's a lot of problems with this.
Actual problems or made-up problems based on lies and cherry-picking?
Duplicated genes are actually often silent. So even if this did occur in the past, that other gene would probably be quieted down.
Probably, so not always. So this isn't a problem if we just don't restrict ourselves to a ludicrously short timeline. Okay, cool, so no problem at all.
Because it's considered a defect a lot of times. Again, in humans, it can lead to a lot of problems.
And it can also not do that. Both of these objections are basically objections that gene duplication does not always lead to new or better functionality. But that's the case for mutations broadly, and it's not new. The question is, can it? It can! So it's
A mechanism.
for long-term evolutionary change. I guess it's back to the good old dice analogy. If you have 120-sided dice, how long will it take to roll them to get 100 of them to be 20s? It would probably take you longer than you have left to live. Even if you are a baby listening to this. But if every time you roll you get to set aside the 20s, then you'll get there pretty damn fast. Similarly, yeah, most mutations, including gene duplications, aren't doing anything or are even harmful. But if any of them aren't, and are instead beneficial, then selection can preserve them, and even some of the just neutral ones will be preserved by drift. And of course, we already know just from dogs that gene duplication that are beneficial can happen within the creationist timeframe.
There's a lot of ways to silence those duplicated genes.
Well, that's true for genes broadly, so yeah. Are they all silenced? No. Are some of the ones that aren't silenced
Actually.
beneficial in some organisms? Yes. So this isn't a problem.
Gene duplication, it turns out, is not a common event.
It doesn't need to be. We have time for it to be a major source of new genes, even if it's not common. Also, it doesn't have to be a full gene duplication to result in new genes.
Now, they did some studies on this, and this comes from the secular scientists.
Okay, so this did not come from monks or nuns, just regular old parish scientists. Okay, jokes aside, what she means is real scientists.
They say they are estimating that there is 0.01 duplications per gene per million years.
I would love to see a citation for that. Not because I think it's not true, but because it would be interesting to read. Anyway, animals have been around for like 800 million years. Humans have, at the low end, around 20,000 genes. So even if we simplify this to a linear rate, we're looking at like 160,000 gene duplications.
Or enough time for all human genes to duplicate 8 times over.
Seems like that's plenty.
That's, now, and I quote, this is what they said in the paper, and I quote,
What paper?
"The origin of a new function appears to be a very rare fate for a duplicate gene," end quote.
Well, that quote was sufficient for me to find the paper she's quoting. It's The Evolutionary Demography of
Duplicate.
Genes by Michael Lynch and John S.
Connery.
published in the Journal of Structural and Functional
Genomics.
in 2003. Unsurprisingly, the quoted bit is from the abstract, because of course it is. In case you were wondering, quoting the abstract is basically de facto dishonest, because the abstract does not contain the conclusion of the paper, and so it's very hard to quote from an abstract and glean the actual conclusion of the paper. Let's just read the whole damn conclusion, which is in this paper labeled as discussion. These results largely corroborate the earlier conclusions in Lynch
Connery.
2000. The estimated half-life of duplicate genes, average over all species approximately 4 million years, is identical in both studies, although it should be kept in mind that there is an order of magnitude range of variation among species. Quote, "The average rate of origin in new duplications in the study, 0.01 per gene per million year, is about the same as that reported in our earlier study." Gu et al. 2002 also recently reported estimates of the rate of origin of duplicate genes in D,
Melanogaster.
C,
Elegans.
and S.
Cerevisiae.
As in our study, these authors employed pre-screening devices to avoid pseudogenes, alternatively spliced variants, and annotation errors, but their additional methods of analysis deviated from those used herein in a number of ways. For example, Gu et al. 2002 did not make a distinction between the number of
Duplicate.
pairs and the actual necessarily smaller number of duplication events. They included very large multi-gene families, whereas we use an arbitrary cutoff for family size of 5. They attempted to remove incomplete or chimeric duplicates, and they eliminated some duplicate pairs in which the nucleotide substitution levels in flanking regions exceeded those in silent sites. Despite these differences, it is comforting that estimates from both studies for B appear to deviate by no more than a factor of 3. Since decisions as to what constitutes a legitimate gene duplication will always involve a certain degree of subjectivity, and since the quality of the current complete genome database is still in a state of flux, there is little question that the rates of birth and death of duplicate genes noted above will be subject to further modification. However, without major upheaval, it appears difficult to avoid the conclusion that ongoing gene turnover is a fundamental evolutionary feature of all eukaryote genomes. Moreover, the smallest genomes may be kept small, not by a low rate of origin of new duplicates, but a high rate of attrition. As a consequence of the duplication process, most species are expected to exhibit
Transient presence/absence.
polymorphisms at multiple
Loci.
see Lynch 2003 for a review, and
Microchromosomal.
rearrangements among closely related species should commonly arise when descendant members of duplicate pairs survive at the expense of their ancestral copy. Such reassignments of
Chromosomal.
locations provide a simple and powerful mechanism for the origin of the post-zygotic isolating barriers that requires no intermediate stage of
Reduced.
fitness and no accumulation of negative epistatic interactions between heterospecific genes, Lynch and Force 2000. Thus, although gene duplication is often regarded primarily as a mechanism for adaptive evolution, its role in the other major engine of evolution, speciation, may be even more significant. Finally, we note that although the majority of duplicate genes appear to be
Transient.
contributing little to long-term phenotypic evolution, the majority of genes become preserved for very long periods, either by subfunctionalization or neofunctionalization, as illustrated by the long, shallow shoulders of the age distributions displayed in Figure 1, and by the strong purifying selection operations on such genes, Figure 5. Although such genes may be simply selected for purely on the basis of redundancy, this appears to be unlikely on theoretical grounds, Lynch et al. 2001. So they almost certainly contribute to adaptive evolution. Nevertheless, unless the eukaryotic genome is undergoing a gradual or prolonged expansion, even members of these pairs must ultimately be subject to loss.
End quote.
Does this say that the gene duplication is too rare to be the source of new genes? Nope. It does say, though, that
There is a more or less
consistent churn of duplicates coming into and out of existence, and that they may play a larger role in
speciation.
than in long-term evolutionary change. But that they do play a role in the latter. It also did not find newly low rates of gene duplication. Also, I'm really annoyed that I had to find this paper by searching for a partial quote of a sentence in the abstract. What is it with creationists and never citing papers? Well, not never, but they seem to struggle to do so consistently.
But yet, that's how we supposedly got all these novel genes.
Nope, gene duplication is not the exclusive way novel genes evolve. That is a straw man, and even Georgia's source doesn't say that it isn't a way we get novel genes.
But it's very rare, okay? It doesn't work. Even if you give it more time, if it's a very rare event, it's still not going to get you to where you need to be.
Did the math, and it actually can. And also, no one thinks it was the only source of new genes. So, even if the math didn't work out, which it did, then so what?
Some genes are actually needed in multiple copies. You need that. So it looks as if it was designed, not evolved, because you need those.
Non sequitur. Your facts are uncoordinated.
Ribosomal RNA genes, for example, are a really good example of that. You need those for the rapid production of ribosomes to get those proteins made. And if you just delete 1 or 2 copies from bacteria, from humans, from flies, they don't do so well. Okay? They need all their copies. So what did they happen in the meantime?
Slower metabolisms, that's what. This is a duplication that probably happened billions of years ago. Life billions of years ago was not the same as life now. It faced much less competition, and so slower processes were fine. There certainly were no fruit flies with metabolisms like a furnace around a billion years ago.
They had to wait for these duplication events to occur so they could survive.
No, they were surviving just fine as things that were
More basal than.
bacteria. In a world where there was virtually no competition for resources compared to today, and so a slower rate of metabolism could be supported by things like slower rates of protein synthesis. Then when these duplications occurred, that probably led to a selective sweep of all the other organisms, which might actually indicate that these duplications happened before the last universal common ancestor of all extant life, or LUCA, even evolved. Because remember, LUCA, Luca, was not the first life form. It had an evolutionary history, and there were contemporary organisms that died without descendants.
I mean, that's a clear problem for evolution, okay? You can't wait around to get all these duplication events so the organism can survive.
Right, but that's not a problem, because the organisms that didn't have the duplications didn't need them to survive. The whole idea of evolution is that organisms change, so that in the past they weren't the same as they were in the present. I guess Georgia was absent that day in postgrad, which is weird, since as a biology major, that's every day in undergrad, graduate school, and postgraduate training. So I guess she just never got to school. Or maybe she's just lying to us about how evolution is supposed to work, because she knows she can't actually argue against evolution in any meaningful way. So she just erects straw men to knock down.
Gene families have very complex interactions, as I told you with beta globin. You know, all of those different globins that are there have different affinities for oxygen, and they're important depending on the developmental status of the individual.
Okay,
Whether they're an embryo or a fetus or, you know, an adult. It varies. And so you need them all, okay? You can't, again, wait around for these duplications to occur because they're needed at different time points throughout the life of an organism.
Except that they're not. We have animals alive today, big, complex animals like alligators and hingas and anacondas that don't need them.
These are a result of and evolved in tandem with the unusual reproductive strategies of mammals.
They weren't needed before that, and their evolution as time went on allowed for further specialization of mammals into their weird, milky, live, birthy nonsense way of making babies. Does Georgia honestly not understand that something can evolve like lungs in a fish and not actually be strictly necessary for survival when it first evolves? Again, like lungs in a fish, but then become absolutely vital to descendants, like lungs in tetrapods? We can see a range of necessity of lungs across different...
Sarcopterygians.
In coelacanths, they're not needed at all, and so have disappeared. In lungfish, they're not vital, but they're handy. In amphibians, they're sometimes vital, like with frogs, and sometimes not, like with salamanders, not all of whom even have lungs. With amniotes, which is a group that contains reptiles and mammals, but not amphibians, they're completely necessary. The same thing can be said for
beta-globin
genes. For animals like reptiles, 1 is fine. For animals like Triassic mammaliforms, perhaps 2 suffice. And for mammals like modern mammals, 4 are needed.
And mutations are degenerative, and we're going to talk more about that in a minute.
Some mutations are harmful, some are not, and unless she defines what degenerative means, I can't really say how many are that. But once again, I really wish she'd just talk about the things she mentions. And if she needs to talk about them later, that she would just mention them later.
They don't add information. They don't lead to something that's getting better and better. It's something that's getting worse and worse. So that's a problem in and of itself.
Well, we don't have a way to measure information that creationists have ever proposed. So saying that they don't lead to more information is meaningless. As to getting better and better, fitness gains from mutations have been observed directly in the wild and in labs, so that's just factually wrong. In the same way that it's wrong to say that Rhode Island is an island country off the coast of Thailand.
So what can we say?
That Georgia,
like most of the rest of AIG,
is a bag of dicks and a liar? I'd say most, because I'm sure that the kids that they hire to run the snack stand at the Ark Encounter are just kids looking for a job. I also happen to have reports from former employees that some of those seasonal workers do things like
see just how much
gay sex they can have on the grounds of the Ark
Encounter.
I, for one, would love if the Ark Encounter became a cruising hotspot. Although I suspect that the exorbitant fees to go there preclude that.
Gene families are not evidence of past gene duplication events.
Nothing that Georgia said even argued for that, except for her not understanding the very basic fact that evolution means that organisms in the past aren't the same as the ones in the present. So her only argument that could work to demonstrate that was just a straw man.
Again, that's a logical fallacy to say that.
Again, empiricism isn't a logical fallacy, it's the very foundation of science, and Georgia knows the difference. She just hates science.
Gene families have complex interactions.
They sure do, at least often.
We simply can't wait around, okay? Organism can't wait around for duplication events to occur.
Yes, they can. They're doing it right now, and that's working out just fine. Sometime in millions of years, I am very confident that there will be an organism that relies on a gene family that right now is actually not needed by its ancestors.
And that gene family exists as a single gene.
In its ancestors. That's how it's always been. Once again, past life wasn't the same as modern life. That's kind of the main takeaway from evolution.
We need them all there at one time, so it really contradicts their gradual formation via evolution.
Yes, we do, but that doesn't mean our ancestors did.
And gene duplication cannot result in the production of new genes with new functions because it's dependent on mutation.
But mutations can indeed lead to new function. One particular notable example is the mutation that allowed a protein in some simian immunodeficiency viruses to evolve the ability to antagonize the human immune protein that is very divergent in humans, allowing that group to become human immunodeficiency viruses, or HIV. As for whether that means that those viruses lost information, well, I don't know how to measure that, so I don't know.
And as we've said before, mutation leads to a loss of genetic information, not a gain of genetic information.
Meaningless. Meaningless. Utterly meaningless. Everything is meaningless.
So it's not going to help you. Even if you have this raw material for evolution to work on, or mutation and natural selection to work on, it doesn't work. Okay? It's not going to get you something new and improved, shall we say.
Except that it very obviously does, and it does it at a taxonomic level that Georgia accepts as one that typically shares common ancestry.
That is, it does it.
At the family, genus, and even the species level. So since Georgia thinks, rather incoherently, that the family is roughly where the kinds are, that means that among kinds, there have been gene duplications and sometimes subsequent beneficial mutations that have in fact occurred after the flood.
So, that's what we can say about gene duplication.
Some really stupid lies and cherry-picked quotes from a single paper. How does one take anything she says seriously?
Now, what about beneficial mutations? This is another really, really popular one. And again, we just got done talking about mutations and what they can and cannot do. So let's focus on that a little bit more.
Sure, let's focus on it. But I see that on screen it says, quote, "Do beneficial mutations add information to the genome?" And I worry
Because that word information
has come up a number of times before and has not been defined. I feel like it won't be defined here either. And I base that on the fact that I have listened to probably hundreds of hours of creationist content and have never found them to define that word and then stick with that definition. The problem is that once they give a concrete definition, either the answer to the question is obviously yes, or it is not really an important question. Because the definition is such that information content isn't a useful metric for a genome, well, let's see what we have.
Do they add information to the genome? Because that's absolutely required if you're going to go from molecules to man.
Yeah, that's true, under any definition I could come up with that could reasonably apply to genomes. At the very least, we could expect human genomes to be larger than those of the first living things, although I'd expect
that of the
genomes of modern bacteria and further modern amoebas and onions both
have
vastly larger genomes than even humans. So by some measure of information, those genomes have much more than those of humans. So perhaps we should ask how much information is needed to go from molecules to onions.
The facts. Beneficial mutations do not exist.
They measurably do, though. Like, we can actually put numbers on it. And they definitely do. Just the Lenski long-term evolutionary experiment with E. coli demonstrates this. Those bacteria underwent mutations that directly caused observed fitness increases in that experiment. To say that beneficial mutation doesn't exist at this point is like saying rain doesn't exist. It very obviously does.
Now, before you all go, okay, what is she saying? Right? The reason I'm saying that is because it's really not the proper way to say that. Beneficial mutations do not exist. However, beneficial outcomes of mutations in specific environments do exist.
Yeah, that's just what beneficial mutation is. Any mutation which causes a phenotypic change that increases the average reproduction
output
of those organisms with the mutation in question in their current environment. So already we're just going to redefine what beneficial mutation is before we even get a definition for mutation. This is going to make me angry, isn't it?
Okay? It really depends on the context whether or not a mutation is a good or a bad thing.
That's true.
And that's included in the definition. This is just standard evolutionary biology, but being presented in a tone that makes it seem like Georgia is correcting something.
And also, with no segue whatsoever,
please consider joining the channel as a member. There's a join link below this video. It starts only at $1.99 a month, US. And you get early access to videos like this, access to my Discord server, and channel emojis, as well as once per month member chat for use on live streams and video premieres.
The most primo example of that is antibiotic resistance in bacteria.
The most primo example? Is Georgia secretly a surfer dude from 90s California? Also, of course I think any beneficial mutation is a good example of a beneficial mutation, but I guess antibiotic resistance
isn't
a bad example.
When there are antibiotics around, that mutation is a good thing. It helps it resist the antibiotic. If you move it into an environment without antibiotics, guess what? It becomes a bad thing. Okay? It cannot compete as well. It's not as fit, so it does worse.
Yeah, that's how beneficial mutations work. The more adapted an organism is to a particular environment, the less well adapted it is to other environments. That's why if we decided to swap out the sun bears to the Arctic and the polar bears to the jungle, both groups would do poorly. So what?
So it depends on the environment whether or not a mutation is a good or a bad thing.
Correct. As far as I know, there is no evolutionary biologist arguing against this.
So there are beneficial outcomes, but not beneficial mutation. I hope everybody's clear on that.
No, I'm not clear on that, because I have no clue what it could possibly mean for a mutation to have a beneficial outcome but not be a beneficial mutation. Beneficial outcomes are what make a mutation beneficial.
Mutations only alter current genetic information. They have never, ever been observed to add genetic information.
If there were ever a time to define genetic information, this would be it.
They can only change what is there.
Well, no, remember last time, they can and also put a whole new copy of a gene, and then that they can mutate into a new gene, and boom, now there's a whole new gene where once there was no gene at all. Granted, that's a two-step process, but still. Further, new genes can arise from junk regions by point mutation that can cause a region to gain an open reading frame. That is a start codon, followed by a bunch more codons, then a stop codon. That's a thing that can and does happen and results in new genes. Further, a change to existing information is still new information. For example, if I had a folder that was full of animation frames, and then I replace one of those frames with a picture of perhaps Luigi, the best Mario character, then that would be new information in the context of that folder. Sure, it might not be a larger amount of information, but it's still new information.
Right? Now keep that in mind.
Okay, I'll keep that in mind.
I have a lot of papers come across my desk of supposedly mutations that have added genetic information. And I've read them all, and I've looked at them all, and never once have I seen one that has added genetic information. They just don't do that.
Wow, this sounds like she has a powerful analytical tool to measure and compare information before and after mutations. I would love to know how she measures information in the genome. I assume there is an actual mathematical model, right? Right? Why do I feel like I'm the Padme and Georgia is the Anakin in the meme here?
Instead, they decrease genetic information. They take away from it. Now, that's not to say that can't be a good thing. In a specific environment, it can be, like I talked about with antibiotic resistance.
Yeah, cool, but I noticed that we still don't know what information is. So I don't know what this means. Like if I said you should watch out because Christianity is false on the grounds that it always decreases Dejorowhack, but according to Christians it's supposed to increase Dejorowhack, the obvious next question would be to ask what Dejorowhack is. How do I measure it? And why is it important in this context? Further, if I then decline to actually explain Dejorowhack, you'd be justified in ignoring me.
But overall, you're degenerating, okay? We're all a bunch of mutants.
No, that's not okay. That doesn't mean anything. It is a tale told by an idiot, full of sounds and fury, signifying nothing.
We're all degenerating slowly over time.
Now that is a claim that needs a citation, especially as life expectancy keeps going up in much of the world, and even places where it's declining, like the former United States, it's still much higher than it was in, say, biblical times.
So that's what's happening. Mutations lead to the decrease in the overall fitness of the organism.
They don't, though, and Georgia has already admitted they don't. Fitness is context-dependent. There's no such thing as universal fitness or something.
May be beneficial in this environment, but not so much over here.
Yep, that's how beneficial mutations work. Look, I'm tired right now. This has been a weird day in a weird week during a year where everything has been on fire. I'm skipping until she makes a new claim that we haven't already heard.
Nylon and nylon-degrading bacteria. How many have heard of this?
Me.
Okay, yeah, quite a few. That is a very, very, very common example that you'll see out there of a beneficial mutation that's added information. So nylon 6, so what's happening here? Well, nylon 6 is a synthetic polymer that was first produced in 1935. But many of us women are very familiar with this because this is hose, okay, that we wear. So that's an important component there of that. And Arthrobacter was found to actually digest the byproducts of nylon 6 manufacture. It was actually breaking down what was left over, so to speak, after they produced the nylon. And since nylon is a man-made product that was only first produced in 1935, then the bacteria could have only recently gained the ability to break down the nylon.
A surprisingly good summary of what happened. Now let's see how the evolution of a brand new digestive enzyme doesn't count as beneficial, or if it does, doesn't count as adding new information, or whatever other dumb shit Georgia's about to tell us.
So, how did they do this?
Well, my memory says that it was a mutation to a different digestive enzyme, but then again, it's not like I have all the papers involved memorized.
How did it gain the ability to degrade nylon? I mean, that seems pretty unique, right? Pretty new.
It is, in fact, quite new.
Did beneficial mutations add information to make these nylon-degrading enzymes? I mean, they didn't have the enzymes to do it, now they did.
I guess that depends entirely on the definition of information now, doesn't it? Too bad we don't have one.
I mean, wow, if that's true, then that would be a pretty airtight case to say that mutations can add information.
It sure does seem that way, yes.
But let's look at this, because they've tried to figure out how they do this. There are 3 enzymes that are involved in the degradation of nylon: E1, E2, and E3. Okay?
This on-screen graphic is why I'm not a big fan of sans serif fonts. Serifs aren't just for decoration.
Those 3 enzymes are absolutely needed. Now, E1 and E3 alter the nylon. They change it so E2 can actually break it down. Now, most of the focus has been on E2, and in fact, they don't even have a mutational analysis yet done for E1 and E3, because people consider E2 more important, because it actually does the degrading part.
I bet we're about to hear her complain about the lack of research, despite the fact that if AIG cared so much, they could funnel some of that ARC money into doing science, this science specifically. Of course they won't, because the more actual science they do, the more it will be obvious how wrong they are, and that would mean that all those donation dollars start to dry up. Of course that's happening anyway, as the economy they helped bring about continues to grind people into the dirt.
E1 and E3 probably have alterations in them as well, but we just don't know about that yet. So we want to take a look at E2, because that's the one people focus on, and that's what you'll see out there when you read about this. E2 is a carboxyesterase. I mean, kind of a big word, but we won't get into all that. It breaks down carboxyesters. That's its role.
Cool. And yeah, I'm mostly just sopping so that the robots that hand out copyright claims don't find me.
Well, they found it had point mutations, single base changes in it, that change the active site of the enzyme. An enzyme is a protein that breaks other things down. And so basically, these carboxyesters will come into this active site, and they will alter them and change them, and then they leave.
Okay, so a gene underwent mutations, allowing for new function in the enzymes produced, which allowed it to create a never-before-seen protein to digest a never-before-seen food. I'm having trouble imagining a definition for information in which this isn't new information. As a fun exercise, see if you can come up with such a definition and put it in the comments.
Okay, well now this active site's been altered. So not only can it degrade carboxyesters, but it can degrade nylon as well. So it can do that. So it still maintains its original function, and it degrades nylon.
I feel like Georgia's doing my job for me.
But what has happened? We've had a reduction of the enzyme specificity.
Yeah? So what? Who cares? When did specificity of the
proteome
become something important to evolutionary biology on its own?
We've lost something. It used to be really specific, right? Used to be specific for discarboxyesters. Now, we've reduced its specificity, and it can do both nylon and carboxyesters. So you've lost something.
We lost the inability to digest nylon in this. Think about that. Does that mean that if an eye evolves, we lost the ability to be blind? Do lungs mean we lost the inability to get oxygen from the air? What the f*** is this? I'm sorry, but there's just no world in which gaining a whole new function and not losing an old one is a net loss. This is unfathomably stupid. My car has lost the ability to sail the ocean. You watching this have lost the ability to not understand English. I've lost the ability to be unable to write scripts about how intentionally dumb Georgia Purdom is. What are we even doing here? This is not a serious argument. How does she even say that with a straight face? And you know that if the E2 gene had a duplication and then one of the copies went back to not digesting nylon, and the other one lost the ability to digest anything but nylon, she'd say that that was a loss of information too, because now the genes have lost functionality. Heads, creationism wins, tails, evolution loses in Georgia's world.
You haven't gained, you've gained the ability to break down nylon, but you've also lost the specificity of the enzyme.
No one cares, that's dumb, and the clown face is appropriate for this.
And this is just a diagram kind of showing you this. Before the point mutations, the carboxyesters will come in, but the nylon can't. Okay, it can't fit in the active site. After the point mutation, both the carboxyesters and the nylon can come in and be modified by this particular enzyme.
Sure, sounds like gain of function to me, and without any loss of function. I don't care about this ill-defined idea of information, but if an organism can gain function and not lose old functions, then I don't see what the barrier is to universal common ancestry that Georgia is pretending exists.
And Dr. Kevin Anderson, who's with the Creation Research Society, and myself wrote a paper on this for the last International Conference on Creationism last year.
That's a prestigious gathering.
Right up there with the most well-attended flat Earth conventions.
And we had this to say about this particular example. Nonetheless, reduced specificity of a pre-existing enzyme is biochemically degenerative to the enzyme, reducing the specificity, even if it provides a presumed phenotypic benefit.
It's not presumed. A particular bacteria was
exploiting
a resource and using that resource to reproduce when no other organism was. That means that they have a new niche and new resources, and so that phenotype will start to increase. That's an obvious benefit with no inference needed.
The beneficial phenotype of nylon degradation requires the a priori existence of the enzyme and its specificity.
Yeah, things tend to evolve from previous things that exist. That's pretty basic to how evolution works.
You've already got to have the enzyme there. You're not making anything new, you're changing what's already there.
A distinction without a difference. Plus, we do know that de novo genes do evolve without having a gene ancestor. This is another example of a common creationist tactic, which is to compartmentalize an aspect of science that they don't like, and then point out that that one aspect does not explain the entire process that they're opposing, and so then declare that the whole process therefore doesn't work. It would be kind of like pointing out that a keyboard isn't a computer on its own as some kind of argument that computing is impossible. Of course, the mutation of genes to create proteins with multiple functions doesn't explain all of evolution. It was never proposed to. It's a single way in which new functions can and do arise, and when you combine it with gene duplication as well as neo-functionalization of previously non-functional regions and subfunctionalization of existing genes, you get mechanisms to gain new functions, new proteins, and for complexity to increase. In fact, subfunctionalization is on its own the loss of some function in a gene, which increases specificity. So, even if we care about that, we have a mechanism that increases protein function specificity anyway. And of course, you know that if Georgia were to talk about some observed process of
subfunctionalization,
she'd just say that it's a loss of function and so it's genetic degradation. But that's the game. All mutations, according to creationists, degrade things, not in any measurable or significant way, just by fiat.
Its degeneration is not a mechanism that accounts for the origin of either the enzyme or its specificity, right?
Yeah, that's other processes.
It's not something that gave you new enzymes. It took enzymes that were already there and altered them.
Wow, like evolution? This just described evolution, but in a whiny tone.
Which is exactly what mutations do. Sometimes it's beneficial, and in this case it is, and we'll talk about more examples of that on Thursday.
Yeah, she might have given another talk around this time, but it's definitely not my next project.
That happen a lot. It's very common in bacteria. But again, it's not adding information, it's altering what's currently there.
Then it's new information. If I took a Bible and replaced a whole chunk of, say, 1 Chronicles with an original story about ancient aliens, that's new information. Sure, depending on your measurement of information, the information amount may have stayed about the same, but it's new information. Similarly, if I decided to print a Bible with the book of Genesis in there twice, and then replace some of that with my story about ancient aliens, well, guess what? That copy of Genesis has new and more information.
By any
Reasonable measurement of information. But of course, by not giving a definition, Georgia can just say anything she likes about information, and it's not like we can say that she's wrong according to her own definition since she doesn't have one.
Now, there's a lot of information out there on the web about nylon-degrading bacteria. Be careful what you read.
Yeah, you might learn some science, and we can't have that now, can we?
A lot of it is not current. It's not based on information that was published in 2007 on this. So anything that's prior to that, please be wary of that, because I found a lot of problems with things that are out there.
Yeah, Georgia rejects the most basic aspects of her own field and the entirety of the idea of science. You should not accept her assessment of what problems may or may not exist in scientific publication. That being said, it is the case that you should tend to prefer newer work in science over older work, since newer work generally can draw from more and better data.
It really shows the amazing adaptability of bacteria that they can do this.
Hey, you know,
What a synonym for adaptability is in this context? Yeah, it's evolvability. And evolvability itself can evolve. That's
why?
some strains of E. coli in the Lenski long-term evolution experiment evolved to become hypermutators. It's because that allows them to evolve faster at the cost of having many low-fitness individuals in the population.
And we'll talk more about that when we talk about bacteria on Thursday. Okay, another famous one you hear about is Richard Lenski and his work with E. coli.
Neat, I just mentioned that. So yeah, you do hear a lot about it.
Richard Lenski is a molecular ecologist at Michigan State University. And he's done some work with the little E. coli bacteria that inhabit our gut. He's cultured 12 lines of E. coli beginning in 1988. Okay? Now that's a long time ago.
For bacteria, it sure is.
And he's had over 44,000 generations of bacteria that have been generated in his lab as a result of that. And some pretty interesting things have come out of that. And every so once in a while, he publishes a paper. And this is what Jerry Coyne, who is an anti-creationist, pro-evolutionist, very much so.
And a sexist and general piece of shit. In case you haven't heard, science, like many aspects of our world, has a bit of a problem with celebrities becoming horrendous monsters. Yeah, he wrote some good papers and at least one great book. He still sucks, and he shouldn't be mentioned without that fact.
Had this to say: Lenski's experiment is also yet another poke in the eye for anti-evolutionists, notes Jerry Coyne, an evolutionary biologist at the University of Chicago. The thing I like most is it says you can get these complex traits evolving by a combination of unlikely events, he says. That's just what creationists say can't happen.
And he might be someone who thinks palling around with the world's biggest sex trafficker and pedo is fine, but
Coin.
is right on this.
Now, are complex traits evolving by a combination of unlikely events?
Yes.
Well, let's take a look at what Lenski found, because you can read his papers and you can see what he indeed discovered.
That's true.
You sure can.
Well, the fitness of the bacteria that are in the lab now is greater than that of the parental strain. The parent bacteria that he started out with, the bacteria has now, are more fit.
After more than 40,000 generations in the same environment, I'd expect so.
Imagine that. They've adapted to life in the lab. Okay, they like it there. But you know what? How have they adapted to life in the lab?
It varies.
Each of the strains has different adaptations. Some have evolved the ability to digest
Citrate.
in the presence of oxygen. Some have become hypermutators. Most have lost their flagella since the flasks are shaken by humans, reducing the importance of propulsion.
They've lost stuff.
And gained stuff. Evolution involves both.
Okay? That doesn't help you. That doesn't add information.
If it didn't help, then it wouldn't increase fitness. As for information, we still don't know how to measure that in a way that Georgia would accept. So maybe it does, maybe it doesn't. We don't have a way to assess.
They've lost the ability to degrade certain sugars.
I don't actually remember that being reported, but if those sugars aren't present in the environment, then it's perfectly plausible that that happened. Just like primates have lost the ability to synthesize vitamin C. Their environment didn't require them to be able to do so, and so when they lost the ability, it wasn't detrimental, and so genetic drift fixed that breakage.
They've lost the ability to move, they've lost their flagella genes, and they've lost certain regulatory controls.
Yeah, that's kind of what you'd expect in such a weird artificial environment. You lose things that you don't need. There's a reason that despite
Having a full suite of olfactory receptor genes,
cetaceans, that is whales, don't have a single functional scent receptor. They don't breathe underwater, so they can't smell their environment, and they only take in air for oxygen, so they don't need air-based olfactory receptors. So over time, they broke. In the meantime, though, many of them acquired complex echolocation systems that give them a whole new sense, and along with it came new proteins not found in other artiodactyls like deer, giraffes, or cows. I bet, though, that Georgia doesn't talk about the lineages that gained the ability to digest
citrate,
in
aerobic
conditions, but then lies about it. Let's see, shall we?
Of course they have. Why should they bother being able to degrade multiple kinds of sugars when they only have one kind they're ever being fed?
Yeah, as evolution would predict. A confirmed prediction of evolution isn't a problem for evolution.
Why should they bother to move? They don't have to move. Food comes to them. Okay? They don't need these things, so they throw them out. There's no point in making them if they never need them. So they've adapted to the lab conditions. That's great. They're more fit in the lab.
Which, is their environment, and adapting to the current environment is basically all of adaptation in the evolutionary sense.
But try putting them in the soil and see what happens.
And try putting an Amazonian tree frog in the South Pole. Suddenly it won't do so well. Of course. Adaptation to an environment is necessarily becoming less equipped for different environments. The fact that organisms aren't adapted to all environments isn't an evidence against evolution. Evolution doesn't say that's even possible, and using it this way is just a straw man.
They die, okay? They don't do very well. They cannot compete with the regular, normal E. coli that's out there.
And if you mix up some wild E. coli in the flask from the Lenski lab with the experimental bacteria, the wild type would also die by being outcompeted. So what?
There's a trade-off. They're more fit for one environment, but less fit for another.
Yeah, that's just evolution. This is high school biology shit.
We already said. So this isn't a poke in our eye.
It sure is, because it demonstrates many aspects of evolution: adaptation, functional gain, functional loss, genetic drift, natural selection.
New genes have evolved, a whole new metabolic pathway
that happens to be irreducibly complex evolved. And whining that the bacteria adapted to their new environment doesn't change any of that, because that's all Georgia has done so far when talking about this experiment.
A feather in our cap, right? It's exactly what we'd expect to see, and that's exactly what we do see. They should adapt.
As predicted by evolution.
And Kevin Anderson and I had this to say: the beneficial mutations, dealing with Lenski's work, the beneficial mutations are very environmentally specific, and a change in this environment often negates the benefit of a mutation.
Which is how evolution works.
When repeatedly cultivated in a constant environment, it is not surprising that an organism would reduce its genome of some unused genes and functions. Why bother with those things? In other words.
Yeah, I agree. It's not surprising that organisms adapt to new environments, because that's what evolution would predict.
Such a reduction can be beneficially selected only as long as the organism remains in that constant environment.
Again, that's how evolution works.
Ultimately, the genetic effect of these mutations is a loss of function useful for one type of environment as a trade-off for adaptation to a different environment.
Well, that's what happened in a lot of cases, but I noticed that Georgia has so far avoided talking about the fact that some strains also added whole new metabolic pathways for
citrate
digestion. Maybe this is before she figured out how to lie about that.
Okay? So, yeah, it's great for over here, but not so great over here. Okay? It's trading off, so to speak.
Again, that's how evolution works.
So evolution, it's not evolution. It's not a gain of information.
Evolution does not require anything to gain information under any definition, and we still don't know what Georgia means by that.
You didn't gain any structures, and you didn't gain any functional system.
All right, guys. Sometimes you are wrong in life.
It's adaptation.
By means of natural selection. The whole thing Darwin was on about. Imagine accepting the very thing that Darwin suggested in the book that created evolutionary biology as a discipline, and then thinking that somehow doesn't actually prove evidence for the very theory it's part of. Will Georgia next say that the bending of light observable as a change in the position of stars during the solar eclipse is actually evidence against special relativity? Will she say that the fact that fires can't start in an atmosphere that lacks oxygen is evidence against oxygen theory of fire? That's the level of twittery going on here.
You're losing functional systems, you're losing information, you're losing structure.
And gaining them in other places.
But it's very specific in relationship to the environment.
I'm going to go insane. Yeah, of course it is, as evolution predicts.
And if you want to read more about this, this paper is located on the Answers in Genesis website.
Which basically makes it trash.
So if you Google, or if you put in our search engine, probably Purdom and beneficial mutations or something like that, you'll get this paper will come up and you can read that for more information. So what can we say? Well, some mutations do provide beneficial outcomes in certain environments.
Which is precisely the definition of beneficial mutation. That means that beneficial mutations occur. Full stop.
That's absolutely true. But mutations have never been observed to add information.
A meaningless statement if we don't have a definition for information along with a way to measure it.
We're looking at the best of the best, okay? This is the best.
I don't know what the best means in this context. The best evidence for evolution, the best mutations, the best lies a creationist who knows better has ever told?
And it's not doing it.
What's the
Antecedent.
For either of the instances of the third person singular neuter
Personal.
pronoun there? Let me know in the comments if you got this as far as what you think that sentence is supposed to mean.
Only decrease information, never add to it.
You know what the response is at this point.
So a trade-off occurs as organisms adapt to an environment. Yes, they're better here, but not so well over here. But the overall fitness is decreasing. They're being becoming very specific for a certain environment.
There is no such thing as overall fitness. Fitness is a measure of the contribution to the genetic makeup of future generations. That's it. It cannot be measured in some hypothetical generalized environment in which a population does not live. This is just made-up nonsense to get around the simple fact that fitness gains are trivial in the real world as expected by evolution.
Beneficial mutations, therefore, cannot be a mechanism for evolution.
She says after describing how they are in fact that.
Because that requires a gain of information. And as we've seen, even with our best examples, they're leading to a loss of information.
That sentence means far less than this one.
So again, starting with God's truth makes sense of what we observe in genetics as it applies to the present and the past.
If there's a God who created the universe is better evidence about that God and the universe itself than any text logically could be. So evidence trumps the Bible when they conflict, and all the evidence is in the camp of evolution when it comes to biology. The Bible has myths about events that we know for certain didn't happen.
And
magic-based inheritance of phenotypes. Sorry, the Bible is wrong.
And that what we observe in genetics confirms the truthfulness of God's word.
Only if by God's word you mean the natural world as we find it. If you mean the Bible, then no, it does not do that in any way. In fact, the evidence says that no matter how many stripy sticks your livestock mates near, it won't have any effect on the stripiness of their offspring.
But starting with man's ideas as the truth, again, leads to a lot of problems that we've seen, a lot of conflicts, a lot of logical fallacies concerning what we observe in genetics and how it applies to the present and the past.
I agree. Starting with man's idea, as found in the Bible, will cause a lot of problems and will make you use a lot of fallacies, as we've seen Georgia do.
And it does not confirm man's idea as truth.
Again, I agree, as long as we realize that the Bible is literally all the ideas of men.
Let's go back to 1 Peter 3:15 here. I shared this with you earlier. We have to be prepared to give an answer.
Nah, let's not go back to her blatant misrepresentation of this verse. Go back to part 1 if you want to hear me address that. The rest of it is just preaching. We never even got to the ability to digest
citrate
in the presence of oxygen in E. coli. Well, Georgia didn't. I did. It was both sad and also kind of vindicating to hear Georgia just say that empiricism is worthless. If you liked this video, please hit like and let me know what you liked in the comments. If you didn't, then hit dislike and let me know what the problem was. Either way, I hope you subscribe and check out the channel memberships and my Patreon. I'm the Dapper Dinosaur.
Hey, before you leave, I just want to take a second to thank my patrons and channel members, especially those pledging $20 or above. Siloof, Benthovend, Tapioca Weasel, Sphincter of Doom, Regios, Landon, Ian Chen, Elleron Teller, Phil Ravara, Kelvin Brostok von Manen, David Hercules, Joshua Geese, Pegajace, The Plastic Mouse, Arrow Dodger, Gordon Love Science, Ember Athelian, Prixie Purple, Agricola Siqua, Me, Myself, and I, Edwin M., Kenneth Swenson, Lance Hall, and John Ackerman. It's because of my channel members and patrons whom you're seeing on screen that this channel can stay afloat. Without you, it would all shut down. If you want to join the team, there's a link to join the channel below this video, and there's a link to join the Patreon in the description. On the Patreon, you can get a 15% discount for pledging annually, and either way, you get early access to virtually all of my scripted videos months before they come out for the general public. Thanks for watching.